we're starting in our hierarchy, right? We started with the atoms, then molecules, now we have the cell. So again, you see the importance of each and every ingredient. You need them all. And if you're lacking, there's anything missing, a deficiency or too much, you suffer the consequences. There's an illness and then eventually will be death. So the cell is the smallest unit of life. That's the smallest unit of structure and function. What is the smallest unit of structure? It's the atom. But structure and function is the cell. So the study of the cell is called cytology. Okay? Look at, we're all trillionaires. We have at least 75 trillion cells. So, finally we have some title to us, right? Trillionaires. And as far as cells are concerned, there are actually two groups of cells. Your sex cells and somatic cells. So, sex cells, there are only two sex cells. That's your sperm and your oocyte or your egg cells. These are the only two sex cells. Not the vagina, okay? Not the penis, because people always mistake that. It's actually just the sperm and the eggs. You differentiate sex cells from somatic cells in terms of how they divide, cell division. But they're all cells. Do you follow? Okay? So everything else is called a somatic cell. Look at the many different types of cells. Look at, you have your red blood cell, your white blood cell, look at muscle cell, and there's more here. You look at your bone cell, they come in different shapes and sizes. But no matter the variety of shape and size, they all generally have the three main parts: the cell membrane, the nucleus, and the cytoplasm. Okay? Cell membrane, nucleus, and cytoplasm. So we're going to look at these three different parts. So first, let's look at the cell membrane right there, the cell membrane. Okay? So, right here. Okay? There's the cell membrane. It's referred to, and that's how it looks like. It's referred to, the composition is made up of, there's your phospholipid, right? The other type of fat. It's a phospholipid bilayer. What is the function of the cell membrane, do you think?
It's to keep stuff in and out.
Yeah, right? So it maintains the boundary of the cell.
It's the guardhouse.
So you know now the term intracellular and extracellular. They're extracellular. You know now that term, right? Intracellular and extracellular. So, because of this, the fat part, the lipid part, substances cannot easily go in and out, right? There's a difference in concentration between substances inside and outside. Inside, in general, or no, all cells have a charge at rest. Does it make sense? Because, you know, your electrolytes, aren't they charged? Your cations, everything is charged, right? Because of the difference in concentration, there must also be a difference in the charge of all my cells. All my cells inside have a negative charge and outside have a positive charge. All cells. So, all cells at rest, this is referred to as a transmembrane potential. There's a potential difference in charge. Inside is negative, outside is positive. Transmembrane potential. You'll see this when we study muscle physiology and nerves also. Okay, you know now the meaning of transmembrane potential, right? Difference in charge. Inside, intracellularly, negative. Most abundant inside is potassium, most abundant outside is sodium. You take that more. But now just know the difference. Because of this one, because it controls the movement of substances, there must be a way for substances to move in and out. There's different processes, right? One, there's two types, the passive and the active movement, right? The passive movement, you have diffusion and osmosis. This is the most common or the easiest way for substances to move from one area to the next, right? So what's the difference between the two? Diffusion refers to solute movement. Osmosis always and only refers to water movement down a concentration gradient, meaning from area of high concentration to low concentration. Let's say I just had my breakfast. Glucose is very high outside. It will enter my cell. How? Diffusion. I drank a lot of water. Water enters the cell. Water follows salt. And that's very important for you to remember. Right? So that's why I want you, and always please show these slides to your friends, to your children, but they don't understand it. But look at this one. This is, all books always use the red blood cell. But remember that every cell is affected by water balance. This is a cell, and this will come out in your test. This is a cell in an isotonic environment, meaning the water concentration outside and inside are the same. So there is no net movement. It's in water balance. Look at the shape, isn't it pretty? Remember that the function of every cell depends on its shape. Look at this cell when it is exposed to a hypotonic solution. Meaning, hypo means too dilute outside. This is when you just drink plain water. So, if you compare then the salt concentration here to here, this becomes fairly salty, right? In a hypotonic solution. Do you follow? So look at the direction of water movement from here to here, right? What will happen to the cell? It will, what? That's right. Called lysis. If it will explode, it's dead, isn't it? This is what happens when we go, like that guy, the Boston Marathon, he's a retired cop. He just retired. To celebrate his retirement, he did the Boston Marathon. But he was so overzealous. He drank and drank and drank and drank water. He didn't finish. He died. I bet you lysis happened. Right? This is irreversible, isn't it? And look at what happens when we expose ourselves to hypertonic solution, meaning too salty outside, like dehydration, right? We don't drink enough water. So at that point, the water inside the cell leaves. Look at the shape. It changed. Yes. It's called crenation. Remember that term, crenation? And lysis, when crenation happens, when lysis. Look at this. Will that, like, this is your red blood, and this is really what happens to red blood cells. Will that be able to do its job, you think? No. What's the job of red blood cells? That's right. So, like if you're dehydrated, there's not enough oxygen being transported because the shape is compromised. Will you be able to concentrate in school? No. So our children, they go home, they didn't do well in school, maybe they didn't drink enough water. That's why it's very important to, Did you know that chronic dehydration is linked positively with cancer? Because what is the definition of cancer? Abnormally shaped cells. Abnormal. Cancer. Right? Especially colon cancer. You see? This is going to come out in your test. Why? To me, it's very important. Very important for you to understand. Your brain, if there's not enough, you just think. It compromises the shape. You will be able to think. Do you have any question on isotonic, hypotonic, hypertonic? Why? Because of this osmosis. It's passive movement. So water just follows wherever the salt is. The other types of movement, active process, means you need energy. You're going against the concentration gradient, meaning where it's already too crowded, you're going from low concentration to high concentration. Active process. You have phagocytosis. So first is endocytosis, meaning entering the cell, right? And the opposite is exo, leaving the cell. And these processes require energy. So this is an example. Phagocytosis is an example of endo, right? Like this is a white blood cell maybe, and it's eating a bacteria. That's called phagocytosis. Fairly large molecules. Pinocytosis is the same. It's phagocytosis, but it's liquid. It's like, Pinot Grigio is like wine. So you're drink fluid. So pinocytosis is also endocytosis. And the opposite is called exocytosis. When you're releasing, like when you just made an enzyme, you want to get rid of it, release it to your GI tract, exocytosis. Okay? Okay, so the next part of the cell that we're going to study is your cytoplasm. So this is the cell membrane. This is the cytoplasm. So the part between the cell membrane and the nucleus is called the cytoplasm. And the cytoplasm is made up of all of these organelles embedded in the cytosol. So cytosol is nothing but the gel-like substance that holds the organelles in place. Okay? So this is the part, it's like the reception area, right? When you ate, you had your breakfast or whatever, when it enters the cell, it goes to the reception area first, the cytosol, the lobby. Then from there, it looks where it should go, then it goes to whatever organelle it's supposed to go. Does it make sense? Okay? Everybody, you think? Okay. So, let's look at the different organelles now, the different organelles. First of all, by the way, write down cilia versus flagella. These are cell extensions, right? They are both for movement, except that the flagella moves the entire cell, and there's only one cell with the flagella. That is the sperm, yes. And the cilia moves substances across the cell. So it's like brush. It moves the substances across the cell, on the surface of the cell. Like in your respiratory tract, all your cells there are ciliated cells. So it brushes up the bacteria that you inhale. So every time you swallow, there's phlegm that we swallow. Okay? But the flagella is for moving the cell itself. Now we look at these different organelles. The first organelle, centrioles. Next to centrioles, you're going to write cell division. It's the key for cells to divide. Okay? So cells with no centrioles cannot divide anymore. What do you think? My skin cells, I'm assuming my skin cells have centrioles because I know my skin sheds, but I still have skin. My skin cells must have centrioles. The bad news is red blood cells, nerve cells, and muscle cells do not have centrioles. So if they get injured, it's either they must have stem cells or you're SOL. Your red blood cells, you have stem cells, right? Your bone marrow stem cells. So you just keep making new red blood cells. But your nerve cells and your muscle cells, that's not the case. So if you had a stroke, the injury is permanent, isn't it? Or like if you had a heart attack, right? You just go for lifestyle changes. There are things you used to do you can't do anymore because when there's an infarct or you had a heart attack, what replaces it is scar tissue, and scar is not muscle. And this is why you need to pay attention when it says in the medication, please, if you experience muscle aches, stop immediately and call your physician. Why? You cannot replace muscle. Okay? Again, why? No centrioles. My students know that by heart, and they remember the 3 cells because I always ask them that. Okay? Next, lysosome. Next to lysosome, write trash can. That's the trash can or the recycle bin. So let's say this is a white blood cell and there's a bacteria, right? And it will eat the bacteria. That bacteria then, it enters the cytosol, then it gets sent here. The lysosome will just break it down to its component parts. After all, it's just made up of organic and inorganic compounds. You recycle it. Does it make sense? Okay, lysosome. Or, yes. So, by the way, we're just talking about the organelles in general. There will be, like your white blood cells, I'm assuming my white blood cells will have lots and lots of lysosome. Not too much of endoplasmic maybe. Does it make sense? Okay. It depends on what it's doing. It will have more of this, not too much of this. Okay, but we're just looking at the general composition of a cell. By the way, write down inclusion bodies. Inclusion bodies are just that, inclusions. So they may or may not be present in a cell. So the function of inclusion bodies, storage. So let's say this cell is storing glycogen, which every cell stores glycogen except for your nerve cells. They store it in globules like that, and if you analyze the composition, it contains glycogen. You call that inclusion body. Or adipose, you call it inclusion body. Same with your melanin pigments, it's stored in an inclusion body. Okay? Next, the next organelle, you write down mitochondria. Next to mitochondria, ATP. This is the powerhouse of the cell. Very good. Very good. And so, remember, I said when we eat our carbs, the bread, it enters the cell. So your glucose, this is where it's sent. When it enters the mitochondria, that is where the glucose is converted to ATP. But what does the mitochondria need to convert your glucose to ATP? Oxygen. So you see, in the absence of oxygen, the mitochondria cannot do its job. I'm assuming my heart must have a lot of mitochondria in my heart muscle cells because it's constantly contracting. Okay? Maybe not too much. It doesn't have centrioles for sure. It does not divide. Okay? Not too much lysosomes. It's not prone to infection. But lots of mitochondria. You see? Okay. Next organelle. You're going to write down your ribosomes. Ribosomes, rough endoplasmic reticulum, and Golgi body. And you make a bracket like this. These three organelles are important for protein synthesis. The bulk of the life of every cell is spent towards making proteins. Enzymes are proteins, antibodies are proteins, your keratin is protein, collagen is protein. All of these proteins that I'm talking about are not just present in the cell. You have to make that when you need it. Okay? So, that's why you have this. Where is the code for the protein? It's here. Remember, the DNA carries the code. You make a copy. The copy is called RNA. Then RNA goes to, the first is the ribosome. In the ribosome is where the protein is being made. And then once the protein is made, it goes to the rough. That's the way I try to remember. You still look so rough. I gotta fix the way you dress up. So this is where it's being modified. Maybe I want to put a little bit of fat over here, some carb over here. That's where it's being done. And then the Golgi body is your FedEx. That's your shipping department. The Golgi body. So the Golgi body will put a stamp. Or are you to be used outside? Okay. Mark for outside use. Or are you to be used by this cell? Okay, I'll put a stamp for inside use. So the protein stays inside. Do you follow? Again, these are the three organelles for protein synthesis. Because all the proteins that we talk about, they're not just magically present in the body. You have to make that. Let's say you're fighting an infection, you need to make antibodies. You have to make that. Where's the recipe? Here, in your DNA. So the first step, you make a copy. You call the copy RNA. Where is that copy going to go? In the ribosome. Here is where you convert or you start cooking and making your protein. Then after that, the protein goes to the rough endoplasmic reticulum. Here, you fix it, make it look pretty. Some of the amino acids maybe not necessary. Take it out. This is where it's going. Then finally, you look good now. Okay, now you're ready to go to the Golgi body because this is your shipping department. Where am I going? You're supposed to go outside. Go to your skin. So, okay, I'm going to go to the skin. Or you're supposed to be used inside here. You're supposed to fight the bacteria that's in here. Then you stay. You follow? Okay? So, those are the three organelles for protein synthesis. Next, you're going to write your smooth endoplasmic reticulum. This is the one that's important for your lipid synthesis, carbohydrate synthesis, and also detoxification. Who has been drunk? Have you ever been drunk? I was drunk. So I'm sure, you heard of tolerance, right? Like if you drink half a glass, you're drunk. Then after a while, 1 glass. Then after a while, 2, 3, 4, 5 glasses, right? This is the reason. Your smooth endoplasmic reticulum makes you tolerant of increasing levels of toxic substances. How? When you drink your alcohol, your cells, your liver cells respond by having the SER neutralize that toxic effects of your alcohol. So the liver cells then start making or producing a lot of this SER. All of a sudden, your liver cells will have a lot of the SER. So you develop tolerance. You drink 2 cups or 2 glasses, you're not as drunk because your liver cells are prepared. They have now more SER. Does it make sense? Same with your antibiotics. This is why, let's say your amoxicillin, 500 milligrams every 8 hours. That's the reason. They consider the pharmacological effect of these drugs in terms of the SER activity. Okay? So this is for detoxification. So if I drink, maybe I'll have more SER than you if you're not drinking. Same with the drugs, right? The cocaine and all that stuff. It's the same. That's why we need to take more and more and more. We become drug dependent. Next will be your peroxisome. Peroxisome sounds like peroxide, hydrogen peroxide. This is also for detoxification, specifically bacteria. So this, when you pour hydrogen peroxide, isn't that supposed to be an antiseptic, right? So this one will kill the bacteria. It doesn't kill our own cells. So to kill the bacteria or pathogens, the product is hydrogen peroxide. The interaction of your peroxidases acting on your bacteria produces hydrogen peroxide. But our cells, our own cells, in addition to containing peroxidase, we also have catalase that converts this hydrogen peroxide to water and oxygen. So our cells don't suffer. But bacteria, they don't have this catalase. So they die. And then, finally, you have what we call cytoskeleton. Skeleton of the cells. Cytoskeleton. These are your the skeleton. The filaments, microtubule, the filaments, tubules. See, look at, they're like the skeleton, right? It lends to the shape of the cell. Okay. So you see the nerve cells, they have pretty shapes. That's because of the cytoskeleton. That's the one that maintains the shape, like our bones. Right? That's why we're able to maintain our shape. Okay? And then lastly, you have your nucleus. What's the function of the nucleus? You know now, right?
Holds the DNA.
Yes. Look at. The DNA is so precious. It's right there in the center. Look at where your nucleus. The nucleus is not to the side. It's in the center, preventing it from being attacked by enemy forces. You have to go through all this before you can attack the nucleus. And look at, what else do I notice with the nucleus? This, the double nuclear membrane, it just is so precious. You need to protect your DNA. Okay? So we formed our cell. Your book has a nice summary, by the way, of the different parts of the cell and the function. I forgot what table. So we formed our cell. We have our organic, inorganic ingredients. I have it now. Very nice. What happens to the cell once it's formed? So once this cell is formed, what is the fate of the cell? The life cycle of the cell. Three things can actually happen to a cell once it's formed, right? It's either, number one, once you form, it can die, right? And there are two types of death: apoptosis, which is your programmed cell death or natural cell death. And the other type of cell death is called necrosis, which is cell death due to injury or abnormal cell death. Necrosis. Like sunburn or gangrene, that's necrotic tissue. That means it died prematurely. Wrinkles, that's premature cell death. Okay? Or it could just remain alive without dividing, right? And in the life cycle of the cell, this is the whole life cycle of the cell. Baby, newborn, infant, adult, becomes old, and he dies. But the bulk of the life cycle of the cell is called interphase, where it's not dividing, it's just surviving. And this is it, interphase. I'm thinking my red blood cells, my nerve cells, and my muscle cells, once they are formed, they remain in interphase until they die. They don't divide anymore. Okay? So you understand they can either die or they can stay in interphase. So what's going on in interphase? Oh, they're busy, right? They're making proteins, they're firing muscle control. They're busy. They're just not dividing. Metabolism is going on in interphase. Okay? Or they will divide. That's the third event that could happen in a cell. They can divide. And if they divide, they will divide by either mitosis or meiosis. And we'll, yes?
The apoptosis, what was that again?
Programmed cell death or natural cell death. Okay? Do you have any questions on the three fates of the cell? So we stop here. We're going to start with our first histology. We're going to do our cheek swab. We're going to look at the cell. And I know most of you have done this when you did your Bio 175. So your first slide, after you're going to do, where is my histology? So what I want you to do is you get a slide, a cheek swab. You're going to swab your cheek and then smear on the slide and put a drop of methylene blue, put a cover slip and look at it. And this is how your cheek swab cells look like. You see, you scrape some of the cells, put it on the slide. Look, now you see there's the nucleus, there's the cell membrane, and then there's the cytoplasm. All the organelles we talked about must be here. Okay? And then, beginning on Wednesday, you're going to see a lot of slides all around you. You're going to start with your histology. You're going to start drawing and looking at your different tissue slides and drawing them under the microscope. Okay? So take out microscopes from there. You can use this microscope. Just take out whatever is on there.
Oh.